Dear students, welcome to the massive open online course on human genetics. Today we are going to learn about the structure of DNA. Let us first begin with the main objectives of this module. To know about the history which lead to the discovery of DNA structure, to know about the types of DNA, to know about the detailed structure of DNA, to know about the various functions of the DNA, and finally to know about the inheritance patterns of the DNA. Dear students, first of all, we will start with DNA. So what is DNA? DNA, deoxyribonucleic acid, is a polymer composed of two polynucleotide chains that coil around each other to form a double helix carrying the genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. DNA is the hereditary material in humans and almost all organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus, where it is called as nuclear DNA. But a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA, or in the chloroplast, where it is called as plastid DNA. It is inherited from the mother to the child. In humans, there are approximately 16,000 base pairs of mitochondrial DNA. Similarly, plastids have their own DNA and they play an essential role in the process of photosynthesis. An important property of DNA is that it can replicate or make copies of itself. Each strand of the DNA in the double helix can serve as a pattern for duplicating the sequence of bases. This is critical when cells divide because each new cell needs to have an exact copy of the DNA present in the old cell. The information in DNA is stored as a code made up of 4 chemical bases like adenine, guanine, cytosine, and thymine. DNA bases pair up with each other, A with T, that is adenine with thymine, and cytosine with guanine, or C with G, to form units called as base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. So DNA is a polynucleotide. Nucleotides are arranged in 2 long strands that form a spiral called a double helix. The structure of the double helix is somewhat like a ladder, with the base pairs forming the ladder's rungs and the sugar and phosphate molecules forming the vertical side pieces of that ladder. Human DNA consists of about 3 billion bases. More than 99% of those bases are the same in all people. The order or sequence of these bases determines the information available for building and maintaining an organism. This is also true for viruses, as most of these entities have either RNA or DNA as their genetic material. For instance, some viruses may have RNA as their genetic material, while others have DNA as the genetic material. The human immunodeficiency virus contains RNA, which is then converted into DNA after attaching itself to the host cell. Apart from being responsible for the inheritance of genetic information in all living beings, DNA also plays a crucial role in the production of proteins. Nuclear DNA is the DNA contained within the nucleus of every cell in an eukaryotic organism. It codes for the majority of the organism's genomes, while the mitochondrial DNA and plastid DNA handles the rest. Now we will discuss about different types of DNA. In addition to the classical structure of DNA, there are several other forms of DNA as well. Let's now discuss the aDNA first. It's a right-handed double helix similar to the bDNA form. Dehydrated DNA takes an A form that protects the DNA during extreme conditions such as desiccation. The major difference between A form and B form nucleic acid is in the conformation of the deoxyribose sugar ring. It is in the C2 Indo conformation for B form, whereas it is in the C3 Indo conformation in A form. A second major difference between A form and B form nucleic acid is the placement of the base pairs within the duplex. In B form, the base pairs are almost centered over the helix axis, but in A form, they are displaced away from the central axis and closer to the major groove. The result is a ribbon-like helix with a more open cylindrical core in A form. In this A form, there are 11 base pairs per turn, 0.26 nanometer axial rise, 28 degree is the helix pitch, and 20 degree base pair tilt. It has a 33 degree twist angle and 2.3 nanometer helix diameter. Now let us discuss bDNA. This is the most common DNA conformation and is a right-handed helix. The majority of DNA has a B-type conformation under normal physiological conditions. bDNA is the Watson-Crick form of the double helix that most people are familiar with. The two strands of the duplex are antiparallel and coiled. The nucleotides arrayed in a 5' to 3' orientation on one strand align with complementary nucleotide in the 3' to 5' orientation in opposite strands. Bases fit in the double helical model if pyrimidine on one strand is always paired with the purine on the other strand. From Chargaff's rule, the two strands will pair A with T and G with C. This pairs a keto base with an amino base, a purine with a pyrimidine. The two hydrogen bonds can form between adenine and thymine, and 3 hydrogen bonds are formed between guanine and cytosine. These are the complementary base pairs. The base pairing scheme immediately suggests a way to replicate and copy the genetic information. It has 34 nanometers between base pairs. It has 3.4 nanometer per turn, about 10 base pairs per turn, and with a 20 angstroms or 2 nanometer diameter of the double helix. It has a 34 degree helix pitch, 6 degree base pair tilt, and a 36 degree twist angle in it. Now let us discuss the zDNA. It was discovered by Andres Wang and Alexander Rich. zDNA is a left-handed DNA where the double helix winds to the left in a zigzag pattern. It is found ahead of the start site of a gene and hence is believed to play some role in gene regulation. zDNA is a radically different duplex structure with two strands coiling left-handed helices and a pronounced zigzag, hence the name pattern in the phosphodiester backbone. zDNA can form when DNA is an alternating purine pyrimidine sequence such as GC, 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 and indeed the G and C nucleotides are in different conformations leading to the zigzag pattern. The big difference is at the G nucleotide, it has the sugar in the C3 Indo conformation like A form nucleic acid, and in contrast to the B form DNA, and the guanine base is in the zinc conformation. This places the guanine back over the sugar ring in contrast to the usual anti-conformation seen in A and B forms of the DNA. The duplex is zDNA has to accommodate the distortion of this guanine nucleotide in the zinc conformation. It is long and thin as compared to bDNA. It has 12 base pairs per turn as compared to 10 base pairs in bDNA, and it has 0.45 nanometer axial rise, 45 degree helix pitch, 7 degree base pair tilt, 30 degree twist angle, and a 1.8 nanometer helix diameter. as compared to 2 nanometer diameter in case of bDNA. Let's now discuss the conditions favoring A form, B form, and Z form of the DNA. Whether a DNA sequence will be in the A form, B form, or Z DNA form, confirmation depends on at least 3 conditions. The first condition is the ionic or hydration environment, which can facilitate conversion between different helical forms. aDNA is favored by low hydration, whereas zDNA can be favored by high salt concentration. The second condition is that the DNA sequence aDNA is favored by certain stretches of purines or pyrimidines, whereas zDNA can be most readily formed by alternating purine-pyrimidine steps. The third condition is the presence of proteins that can bind to the DNA in one helical conformation and force the DNA to adopt different conformations, such as proteins which bind to bDNA can drive it to either A or Z forms. In living cells, most of the DNA is in a mixture of A and bDNA conformations with a few small regions capable of forming zDNA. Dear students, in addition to aDNA, bDNA, and zDNA, there are other rare forms of DNA also, which are cDNA, ddNA, and eDNA. So let us first discuss the cDNA. It's a right-handed with the axial rise of 3.32 angstroms per base pair, and there are 9.33 base pairs per turn. Helical pitch is 3.32 angstroms into 9.33 angstroms, and a base pair rotation is 38.58 degree, and it has a diameter of 19 angstroms, smaller than that of the aDNA and bDNA. The tilt in this DNA is of 7.8 degree. Let's now discuss the ddNA. This rare variant has only 8 base pairs per helical turn. These forms of DNA found in some DNA molecules devoid of guanine. The axial rise of 3.03 angstroms per base pairs and the tilt of 16.7 degree of the helix. Now let us discuss the eDNA. eDNA is also called as extended or eccentric DNA. eDNA has a long helical axis rise and base perpendicular to the helical axis, deep major groove and the shallow minor groove. So eDNA allowed to crystallize for a period time longer, the methylated sequence forms stranded aDNA. eDNA is the intermediate in the crystallographic pathways from bDNA to aDNA. Dear students, before moving ahead, we will focus on the discovery of the DNA. Many people believe that only American biologist James Watson and English physicist Francis Crick discovered DNA in 1953. But actually, the landmark discovery of double helix structure of DNA by Watson and Crick relied heavily on the work of other scientists. DNA was first identified in the late 1860s by Swiss chemist Friedrich Mischer. Then in the decades following Mischer's discovery, other scientists, notably Phobos Levine and Erwin Chargaff, carried out a series of research efforts that revealed additional details about the DNA molecule, including its primary chemical components and the way in which they joined with one another. Chargaff's realization that A pairs with T and cytosine pairs with guanine, combined with some crucially important X-ray crystallographic work by English researchers led by Rosalind Franklin and Maurice Wilkins, contributed to Watson and Crick's derivation of the three-dimensional double helical model for the structure of DNA. Watson and Crick's discovery was also made possible by recent advances in the model building or the assembly of possible three-dimensional structures based upon known molecular distances and bond angles, a technique advanced by American biochemist Linus Pauling. Without the scientific foundation provided by these pioneers, Watson and Crick may never have reached their groundbreaking conclusion of 1953 that the DNA molecule exists in the form of a three-dimensional double helix. Watson and Crick proposal of the double helix model of DNA was actually putting the evidence together like a jigsaw puzzle. So using cardboards, cutouts, representing the individual chemical components of the four bases adenine, guanine, cytosine, and thymine, and other nucleotide subunits, Watson and Crick shifted molecules around on either desktops as through putting together a puzzle. They were misled for a while by an erroneous understanding of how different elements in thymine and guanine, specifically the carbon, nitrogen, hydrogen, and oxygen rings, were configured. Only upon the suggestions of American scientist Jerry Donohue did Watson decide to make new cardboard cutouts of the two bases to see if perhaps a different atomic configuration would make a difference. Not only did the complementary bases now fit together perfectly, but that is with adenine with thymine and cytosine with guanine, but with each pair held together by hydrogen bonds, but the structure also reflected the Chargaff's rule. The double helix structure of DNA molecule was later discovered through the experimental data by James Watson and Francis Crick. Finally, it was proved that DNA is responsible for storing genetic information in living organisms. Although scientists have made some minor changes to the Watson and Crick's model or have elaborated upon it since its inception in 1953, the model's four major features remain the same yet today. These four features are as DNA is a double-stranded helix with the two strands connected by hydrogen bonds, and the bases are paired with each other as per the rule, that is thymine is always paired with adenine and guanine is always paired with cytosine, which is consistent with and accounts for Chargaff's rule. Most DNA double helices are right-handed, that is, if you were to hold your right hand out with your thumb pointed up and your fingers curled around your thumb would represent the axis, the helix, and your fingers would represent the sugar phosphate backbone. Only one type of DNA called the Z-DNA is left-handed. The double helix is anti-parallel, which means that the 5 end of one strand is paired with the 3 end of its complementary strand, and the vice versa. Further, nucleotides are linked together with each other by their phosphate groups, which bind the 3' end of one sugar to the 5 end of the next sugar. Not only are the DNA base pairs connected via hydrogen bonds, but the outer edges of the nitrogen-containing bases are exposed and available for potential hydrogen bonding as well. These hydrogen bonds provide easy access to the DNA for other molecules, including the proteins that play vital roles in the replication and expression of the DNA. Now, we will learn about the fine detailed structure of the DNA. The DNA structure can be thought of a twisted ladder, described as a double helix. It's a nucleic acid, and all nucleic acids are made up of nucleotides, which are the building blocks of DNA. The basic building block of DNA are nucleotides, which are composed of a sugar, a phosphate group, and a nitrogenous base. The sugar and the phosphate groups link the nucleotides together to form each strand of DNA. Adenine and thymine, guanine and cytosine are four types of nitrogenous bases. These four nitrogenous bases pair together in a way that adenine pairs with thymine and guanine pairs with cytosine. These base pairs are essential for the DNA's double helix structure, which resembles a twisted ladder. The order of the nitrogenous bases determines the genetic code or the DNA instructions. Among the three components of DNA structure, sugar is one which forms the backbone of the DNA molecule. It also called as the deoxyribose, and the nitrogenous bases of the opposite strands form the hydrogen bonds, forming a ladder-like structure. The DNA molecule consists of four nitrogenous bases, as already discussed, that adenine, thymine, guanine, and cytosine, which ultimately form the structure of a nucleotide. The A and G are purines, and the cytosine and thymine are pyrimidines. The two strands of DNA run in opposite directions. These strands are held together by the hydrogen bond that is present between the two complementary bases. The strands are helically twisted, where each strand forms a right-handed coil and 10 nucleotides make up a single turn. The pitch of each helix is 3.4 nanometers, hence the distance between two consecutive base pairs, that is the hydrogen bonds bases of the opposite strands, is 0.34 nanometers. The DNA coils up, forming chromosome, and each chromosome has a single molecule of DNA in it. Overall, human beings have around 23 pairs of chromosomes in the nucleus of the cells. So DNA also plays an essential role in the process of cell division. Specific sequence of nitrogenous bases that code for particular protein or regulatory RNA molecules are called the genes. Each strand of DNA is like a recipe book for synthesizing proteins. Certain sequences of nitrogenous bases along the strand encode particular RNA molecule. So these sequences are called genes. mRNA molecules transcribed from genes are translated into proteins later on. Chromosomes can vary widely in their number of base pairs and genes. The longest chromosome in human cells is chromosome 1, which has around 249 million base pairs and has between 2000 and 2100 distinct genes, whereas the chromosome 21 is the shortest among the human chromosomes, consisting of only 48 million base pairs and contains between 200 to 300 genes. Overall, prokaryotic cells have shorter chromosomes with fewer genes. For example, the bacterium Carsonella radii has only 1,59,662 base pairs and 182 genes in its entire genome. Although genes get most of the credit for what DNA does, they make up only about 1% of the human DNA. Genes are separated from one another by sequences of nitrogenous bases that don't provide instructions for RNA synthesis. These are called intergenic regions. Even within genes, there are regions of non-coding DNA called as introns. So non-coding regions of the DNA are important because they provide binding sites for proteins that help activate or deactivate the process of transcription. They can also provide protection for the coding regions. For instance, telomeres consist of repetitive sequences that protect the genetic information on each DNA molecule from being damaged during cell division. Dear students, we will now study the important bits of the final structure of DNA. Note that the two chains run in opposite directions and the right-handed chain is essentially upside down. If you followed the left-handed chain to its very end at the top, you would have a phosphate group attached to the 5 carbon in the deoxyribose ring. If you followed it all the way to the other end, you would have an hydroxyl group attached to the 3' carbon. In the second chain, the top end has a 3' carbon and the bottom end has a 5' carbon. This 5' to 3' notation becomes very important when we start talking about the genetic code and genes. The genetic code in genes is always written in the 5' to 3' direction along a chain. It is also important when we take a very simplified look at how DNA makes copies of itself. Dear students, we will discuss in brief the functions of the DNA. DNA is the genetic material which carries all the history information, and genes are the small segments of DNA consisting mostly of 2 to 250 million base pairs. A gene codes for a polypeptide molecule where 3 nitrogenous bases sequences strands for 1 amino acid. That is, the genetic code is triplet, where 3 bases code for a single amino acid. Polypeptide chains are further folded in secondary, tertiary, and quaternary structures to form different proteins. As every organism contains many genes in its DNA, different types of proteins can be formed. Proteins are the main functional and structural molecules in most organisms. Apart from storing genetic information, DNA is involved in replication process, transferring the genetic information from one cell to the daughter cells and from one generation to the next, and equal distribution of DNA during the cell division. It's also involved in mutations, the changes which occur in the DNA sequences, transcription, cellular metabolism, DNA fingerprinting, gene therapy. The DNA is called a polynucleotide because the DNA molecule is composed of many nucleotides which are combined to create long chains called a polynucleotide. As per the DNA structure, the DNA consists of 2 chains of polynucleotides. Dear students, it was all about today's lecture regarding the structure of DNA. Hope you have understood it well. Take care. Goodbye.